Hello, happy summer solstice, everyone. Thank you for joining us today for the webinar. My name is Jason Key. I'm at Harvard Medical School in SB Grid. And today we're lucky to have Ivet Bahar joining us from Pitt. So Ivet, I skimmed your CV. You started out in chemical engineering, it seems, and then made the leap to computational chemistry at some point. And uh, and then moved to Pitt, and your lab has developed a number of software tools. And today you're going to tell us about elastic network models and ProDy. Is it ProDy or ProDy?
D.
ProD? ProD. Okay, good.
This is the way we pronounce it.
Yeah, great. So whenever you're ready, go ahead.
Okay, thank you very much. I would like to talk about, as Jason just mentioned, the elastic network models that our lab introduced, and also the application programming interface ProD. ProD comes from protein dynamics. Essentially, both the model and the interface were developed for analyzing bimolecular systems. Adopt a network representation. And here on this slide, I'm just trying to say that network models are very useful for several reasons, and here are three of them. We may, having network models, we can see how large systems undergo cooperative motions. We can view long time processes that are much beyond the capability of simulations. We can incorporate structural data in the models at multiple levels of resolution and relate the overall structure to function and allostery. And finally, we can take advantage of the theories that have been developed in other disciplines, like polymer physics theory. And in fact, my background is polymer physics and engineering, so that I've been using that background to introduce the elastic network models for proteins about 2 decades ago, in 1997, the first paper. So the whole idea is the following. The proteins are not static, as we know. They move, breathe, they work, they dance, if you like, and they interact with each other. Here I'm showing a molecular chaperone from bacterial chaperone Groyes, some of you may be familiar with. And using ProDy or network models, it is possible to have an understanding of what is happening collectively in the molecule under equilibrium conditions, under physiological conditions. And you can see, for example, as shown in this movie, you can see the interfacial dynamics, which is often important for determining explain later on in more details. Or alternatively, you can simulate also using ProDy the machinery, the global machinery of the entire molecule. And so this is an allostery machine, ATP. We'll see how the structure changes during its allosteric cycle. These are types of cooperative events that easily and here are a few examples just to give you a flavor. On the left, virus, satellite tobacco mosaic virus, the structure of which, including the viral capsid and the genetic material, has been resolved. So what we are essentially doing is to take the PDB result structure, map it into an elastic network model representation, and then analyze what its most probable are. And on the left, you see the capsid. On the right, there is a subunit from the Groyad, again showing how the structure reorients itself upon ATP binding, for example. And this is the last example that is an AMPA receptor. And for that structure, this is a tetrameric membrane protein. And it is possible, again, to predict the collective motions and make some testable predictions. In this case, we hypothesize that the residues on top here, which are separated by about 30 angstrom under equilibrium conditions in the X-ray structure, we said that they have this ability to come into close proximity and even form, for example, disulfide bridges if we substitute cysteines there. And it was experimentally indeed verified. But not only that, the experiments also showed that lacking these type of movements impaired the signaling properties of the AMPA receptor in a study that has been recently published. So the bottom line is that we are able to predict this type of very cooperative events that are uniquely encoded by the structure. And these are to come up with testable hypotheses about the mechanism of function. So these are this is just a general overview of ProDy. Now let me talk a little bit about what we are doing. Actually, we have a high performance computing biomedical technology research center on multi-scale modeling of biological systems, joint between the University of Pittsburgh, Carnegie Mellon University, Institute for Biological Studies, and the Pittsburgh Supercomputing Center. And the overarching biological theme is to understand the spatial organization and temporal evolution of synaptic signaling and regulation, IMS. So to this aim, we are considering structure at multiple scales. Uh, and this comes from brain regions all the way to single molecules or machines like the CAMK2 here at the panel G. So you can see different time scales. In our center, we are essentially building models for analyzing the dynamics of all these systems. Indeed, some of the images are simply reconstructions, in silico reconstructions. In panel C, we have the dendrite and the organelles, the mitochondria and ER, as well as panel D. So we are essentially using structural data for understanding the dynamics of these biomolecular systems using basic physical chemical principles. And if you go to the website, you can find more information on Bios, the type of studies that we are performing. So at Bios, we essentially have two most important tools, among others. And these tools are, one of them is the one I'm going to describe today. And the second is Amcell, which is a Monte Carlo simulation of cellular processes using detailed structural information on the cell. And by using just this type of reconstructed structures, we are able to simulate the central nervous system signaling and regulation events with spatial complexity and stochasticity. So let's come back to ProDy, which will be the major topic of today's talk. ProDy has been introduced originally in 2011, and then we had a new version again published in 2015. Among the molecular resources that we are using, I would like to cite three, most important. One is the anisotropic network model web server that we have. Again, a new version has been published last year, as you can see here from the reference. So the short name is ANM server. I would strongly suggest you to try ANM. It's extremely easy to use. You just need to enter the PDB code of your favorite protein or system, and it's gonna generate animations, movies, the collective motions. It's designed for people who are not necessarily computationally skilled. You can retrieve outputs very easily, or movies. So that's something I strongly, encourage biologists to do. And indeed, ANM is very broadly used by in the biological community. Another resource that we have is the Gaussian network model database, which also has been updated very recently, as you can see from here. I will talk about what the Gaussian network model is about, but again, this provides you with information on coupled domain movements, the most critical residues, and the types of correlations between movements. It's very, again, in this case, we have about 95% of the PDB structures are stored in the database, including the largest ones. And it is very easy to retrieve the data and analyze the result. I would also strongly encourage you to see what you can do, what type of information you can get. You can obtain fluctuation spectra, you can obtain cross-correlation maps. And what is beautiful is that everything is being released for the biological assemblies. So in some cases, you may be familiar, the PDB just gives you the coordinates of an asymmetric unit. So the database has been constructed to generate the entire, intact structure and release information on biological assemblies. And finally, the third most important is ProDy. ProDy is different a little bit. It does the same. It has the same features, but it is an application programming interface. That is, it has an open source code, and anyone can contribute to it. It's written in Python, and it keeps growing. It has multiple features. Here's a description of how it started. So originally we said we would like to compare experiments and theory and see whether the motions that we predict for bimolecular structures, whether they make sense, whether they are functionally meaningful. So one way to test that was to compare the alternative conformations that we are generating to those that have been already resolved for a given protein. So, for example, suppose you have a given protein sequence. Okay, you can input the protein sequence to ProDy, and it's gonna then retrieve from the PDB all the structures that contain this sequence. This may be a given enzyme, for example. In this case, we are showing for P38 kinase. So P38 kinase may be resolved in multiple different forms in the presence of inhibitor or in under different crystallization conditions. So we have, or by NMR. So we have essentially in the PDB an ensemble of conformations for that particular kinase. So what we're doing is to take that ensemble and subject it to principal component analysis to extract from this experimental data the most dominant modes of motions. I'm sure many of you are familiar with principal component analysis, so I'm not going to go into the details, but essentially, we are reducing this complex ensemble of various conformers into a we are projecting them, if you like, into a reduced space where we can visualize the most prominent, the most important changes in structure. On the other hand, using the ANM, it is possible also to predict the alternative conformers using elastic network model. And then we can compare the principal modes from experiment versus the ANM modes from the theory, and see how they overlap in this overlap table. So that type of comparison has been showing again and again that the structural variations that we observe for a given protein are actually nothing else than what could have been predicted using a single structure. So those alternative forms, the mutants, bound or unbound forms, different states during an allosteric cycle, they are all conformations that are actually easily accessible. We call them soft modes of motions. So it's possible to predict them. That's very important because that way the structure relaxes under different perturbations. That's being used in SB grid on deformational elastic network used for refining structures, because the structures will be rearranged along their soft modes that are predicted by elastic network models. So it is possible to sample many conformations, compare them with MD. And what is interesting, I would like to emphasize, the ensemble of conformers that we generate by ANM, the anisotropic network model, actually provide a better description of the space of conformations observed in experiments than those sampled during MD simulations. MD simulations may be just sampling very local events, or can just go in one direction, be trapped around a minimum. So in the case of elastic network models, we don't have such problems. The only essential aspect of it, which is its simplification. We don't have full atomic resolution. We have an understanding of the overall shape of motions, okay? The backbone, essentially. Here are actually two students. Ahmet has been really the pioneer behind all this effort, and Anindita, she graduated more recently. She was involved in the second version. Actually, this is It's not impressive. It's published now. And a few other people from my lab, Tim and Chakra, made major contributions. They are both faculty members. And Yingiliu is a former student, and Jihan, a current student. They are all making active contributions to ProDy development. These are the key people. And the references again, if you would like to see, they are both in Bioinformatics 2011 and 2014. Just to give you an idea, the usage and dissemination statistics, you can see that although we just launched the interface in November 2010, in less than 6 years, we have had almost, 500,000 downloads or 50,000 unique visitors from all over the world. And we are really very pleased with the overall access to our interface. Here are, and it's a very well documented interface. You can see lots of information on the tutorials. Again, ProDy was the core. We have the visualization interface, which is actually an NMD plugin for those of you who are familiar with VMD plugin, VMD, the visualization software that is used by NAMD, MD package. We have a interface which is provides information on sequence conservation and compensating mutations. And the sequence correlation coevolution patterns can be compared to structural correlations. So ProDy provides this type of comparisons. And another module helps perform drugability simulations. So it has really many different aspects. Now let me go a little bit back to the theory, where everything is coming from, and explain more a bit the technical details. So actually, everything is, as I mentioned, I am coming from polymer physics, and thesis on polymer physics actually, and I was very the theory of Paul Thorey, the 1974 Nobel Prize in Chemistry, on rubber elasticity has been a major inspiration, which led to conceptualizing the elastic network models for proteins. Here is a brief summary. Instead of the usual ribbon diagrams, we use this type of network models. And essentially, we are coarse-graining the structure and the energy surface in a sense, which facilitates the collective movements near the energy minimum and permits us to visualize collective changes in structure. So here is a movie that a student of mine prepared for describing the anisotropic network model. Essentially, we consider the backbone, as you can see. The alpha carbons are the network nodes, and we connect the alpha carbons by elastic springs. So here is a network representation of adenate kinase. Then, having this network representation, we use physics. We evaluate the different modes, normal mode 1, 2, 3, as you can see here. So the structure has a bunch of intrinsic modes that are favorable, and we can predict them by the theory. So for a review, you can, for example, see this review published in 2010. And that one is probably good enough. And I mentioned two elastic network models. One of them is the Gaussian network model, GNM, and I'm just giving here the most recent reference and the original reference. The original was in 1997, published in a journal that doesn't exist anymore, Folding and Design. However, this is my most widely cited paper, more than 1,000 citations. And the anisotropic network model, again, originally published in 2001, and most recent server version has been published in 2015. Those of you who would like to see the original reference. So let me give a more details on the Gaussian network model. So as I mentioned, we are reconstructing the structure based on beads and springs. We use those position vectors Ri for residue positions. Especially what we are evaluating is the changes in the position vectors. And if we take the difference between two of them, we obtain the change in the distance vector. So the change in the distances or in the changes in instantaneous positions during the thermal vibrations, equilibrium motions of bimolecular structures are, to our view, essential to performing their function. And among the different types of motions, some of them are very cooperative, uniquely encoded by the structure, and these are the ones we are trying to elucidate. We were inspired by the Rouse model for polymers, which is like a bead and spring model, but and you can represent the connectivity by what's called a Kirchhoff matrix. You have zeros everywhere except for this band along the diagonal, and which just describes the fact that 1 is connected to 2, 2 is connected to 3, 3 to 4, etc. And the diagonal elements are simply the number of connections at each bead. So the first one has one connection, the second has two, etc. Now, for this system, you can write the total potential as a sum of harmonic potentials, changes in internode distances, and it is possible to rewrite total potential as a product of three matrices based in terms of fluctuation vectors and Kirchhoff matrix. So you can use you can write the total potential in such a very compact notation. And indeed, we use exactly the same notation for proteins, with only the major difference that the Kirchhoff matrix now has non-zero elements in the off-diagonal terms because we have those non-bounded contacts. But this you have lots of non-zero terms on the non-diagonal regions. Uh, so this matrix provides a complete description of the contact topology, and it may be shown, I'm not going to go into the details, but it may be shown that the cross correlations between the fluctuations of residues may be obtained from the inverse of the Kirchhoff matrix. Ijth element will describe the correlation between and residues, or the ith diagonal element will give you the mean square fluctuations of the ith residue. Here is the application to hemoglobin, where we accounted for described by the B factors, or we can obtain the cross correlations between the residues. We can observe that we have like a dimer of a dimer structure, which is typical of the tetrameric hemoglobin. So one little remark before we proceed further. Although we've been using B factors in the very beginning for verifying the predictions, B factors themselves are affected by crystal contacts. So for a given protein, a crystal structure, contacts, there are those contacts between adjacent replica, which of course do not exist under physiological conditions in solution. And the B factors in those cases will be naturally more reflecting the fact that there are such intermolecular constraints. So the corresponding residue fluctuations would be suppressed. And indeed, when we compare theory and experiments, the fluctuations as a function of residue number, the, we found that the theory really, there is a discrepancy at those regions between the theory and experiments. We wanted, we got practically the same results as the B factors. So in a sense, the theory became equally wrong, if you like, because in reality, we don't have such contacts. But this is to say that the theory is in general providing a very good description of what's experimentally observed. More important is the fact that we can decompose the structure into a superposition of normal modes. And I'm going to go a little bit faster here. But as I mentioned, we have the cross correlation given by the ijth element of the inverse of the Kirchhoff matrix, of course, multiplied by a proportionality constant. Gamma is the uniform force constant that we use for all springs. Now, it is possible to decompose by eigenvalue decomposition. You can write this matrix as a sum of matrices, each weighted by this inverse eigenvalue and represented by the eigenvectors. So in this way, we can extract the contribution of the slowest or softest modes, and we can that is very important because now we are removing, if you like, the high frequency motions and focusing on pure most cooperative events, and we have been correlating these things to function. So just to show you the difference, if you extract the slowest mode, here's the profile, the mobility. The okay. And these peaks will be corresponding to regions that are subject to very high concentrations. And we have, if you look at the energy landscape, the slowest or softest modes are nothing else than the motions along the directions that are easiest to undergo. So, in summary, we take the structure, we convert into a network representation. Using the coordinates of the nodes, we construct the Kirchhoff matrix, connectivity matrix, and the inverse of the connectivity matrix gives the correlations as well as cross correlations. And then you can also decompose it into the contribution of individual modes to observe dominant patterns. Uh, so that is the overall summary. And here's the recipe. Actually, you can find this type of information on the web, on our GNM site or in ProDy. So I'm gonna move to the next topic, which is, oh, and I would just recommend you to really try iGNM, the GNM database. You will, and if you find any bugs, of course, please report. But in principle, it seems to be very useful. Here is the address: internet GNM at ccbbpit.edu. So why do we use this database, IGNM? Because it provides easy access to pre-computed results for 95% of the PDB, including the largest structures beyond the scope of MD, the protein, DNA, RNA complexes, and the biological assemblies in their intact functional states. And you can, there are lots of visualization interfaces, so you can you can easily see the couplings, the different domain movements, etc. In the case of the anisotropic network model, ANM, this is like the three-dimensional extension of the GNM. So again, we have the structure, we convert it into a network model, and then we construct, instead of the Hessian, now we instead of the Kirchhoff matrix, we construct now the Hessian matrix, which is found from the second derivative of the potential. And it is possible to generate a series of modes of motions, which I'm showing here. For example, in this case, mode 1, as indicated by the arrows, is like an overall stretching, opening of the cleft between the two domains. Mode 2 is some kind of a torsional motion. Mode 20, a much higher mode, shows some localized motions. And schematically, the corresponding energy cost for mode 1 is relatively smooth surface. In the case of higher modes, it's very steep. So for information, in you can write potential as a sum of harmonic interactions, changes in interresidue distances, quadratic functions, and you take just those residues within a cutoff distance of RC, usually taken like 12 to 15 angstrom. And from the second derivative of this potential, you generate the super elements of the Hessian matrix. Super elements mean 3 by 3 matrices. Okay, and this 3 by 3 Hessian now NM replaces the Kirchhoff matrix of the GNM to yield the mode shapes in a 3n dimensional that one is the name is a little bit bulky, the internet name. Uh, However, if you just type A&M server in Google, you this will be practically the first thing you will see. And again, it's very easy to use. Just enter the PDB code of your favorite molecular system, and you can visualize the results and save the data. The major utilities are for analyzing allosteric changes in conformation. Uh, for example, when you apply to HIV protease, the server will give you this type of color-coded diagrams in addition to their movies, the red regions being the most flexible regions. For those of you who are familiar, HIV reverse transcriptase has a finger domain and a thumb domain, and these are really very flexible. And essentially, we are starting from an energy minimum here at the center of the energy landscape. And when we deform the structure along the A&M modes, we are viewing alternative substates, which we call S1, S3, etc. S1 is the starting point, S2, S3 are alternative substates, which may have functional significance, as I will explain. One utility of the A&M server is to generate an ensemble of conformers, which may then be used for docking simulations. Because as you can see in this particular example, you may have alternative structures, of course, in the presence of different inhibitors. And as you probably know, doing rigid docking simulation are not good enough because proteins are not static themselves. So it is possible, one way of overcoming this type of complexity is to start the simulations with alternative conformations that may be easily generated using the A&M server. So let us see an application to GROIA again, the example I showed in the beginning. So GROIA, here are the two different conformations of GROIA, the R and T states. And I'm just focusing on two subunits here in the two respective conformations. And I'm showing the two subunits here. So as you can see, there is a substantial structural change between the two. Now, suppose now I'm examining this structural change, and I would like to see whether the experimentally observed structural change is well described by the AI. So quantitatively, I predict what this structure is, for example, for overall complex. It's the beauty of the GNM and calculations is that you can do these things within seconds and at most minutes in the largest systems. So ANM will give you a series of 3n-dimensional deformation vectors, different modes, which describe the vector respective frequencies. Now I would like to see whether the experimentally observed deformation agrees with the softest, easiest modes of motions that the structure encodes. So these easiest modes of motions are the A&M soft A&M modes that we would like to see that those soft modes indeed agree with experimentally observed changes. And here is the result for that particular system. You have the mode index and how much each mode correlates with experimental change. So you can see that in this case, the first mode yields a correlation of about 0.8. That this one is a spectacular result. So essentially, what is being experimentally observed between the T and R states of GROIAL is nothing else than the rearrangement of the entire molecule along with intrinsically encoded easiest, softest mode of motion predicted by A&M. So that is one way of verifying that those modes are meaningful. And then we can also do analyze the effect of mutations. That's although intrinsically we don't have residue specificity in the elastic network models, we can observe what type of changes in interactions are induced by the global motions. For example, this at the interface, we see that the rearrangement of residues is very likely. So the molecule in its softest mode, mode 1, has a tendency to undergo this type of movement. And then in the presence of mutation, actually, which would stabilize the native state, the molecule can easily transition to this alternative state, and that's experimentally what has been observed indeed. So the bottom line is that mutants, alternative conformers observed in mutants, are also usually along those motions that are intrinsically accessible. That is one way of predicting how a mutation might affect the structure. And we have also developed methodologies that combine A&M analysis with MD simulations. And so that is also one of the modules. That is a very powerful method because A&M predicts the most cooperative directions of motions at a coarse grain scale. But now we can fill the details with the help of MD simulations, and we can construct the energy landscape projected onto the principal components. Now, instead of comparing two endpoints, which I just mentioned, the R and T states, a better comparison would be to take the large ensembles of structures and to see whether the alternative conformations sampled by A&M represent the ensembles that have been observed experimentally. So here's an example for HIV reverse transcriptase again, an example of alternative conformations. So from those known structures, we already have an idea of what the motions are, but we can then systematically analyze them. We can analyze the structural changes that accompany protein binding or those induced by ligand or inhibitor binding, or those that are visited along an allosteric cycle. So if we give a protein, we have this type of ensembles of conformations, and we perform an ensemble analysis. So what is it? We take as input those structures, NMR models, X-ray structures, resolves under different conditions, or you can even take MD snapshots, frames. And then we obtain we try to visualize the principal modes of conformational variations between the NMR models, between the X-ray structures under different functional states, or even the principal dynamics of MD trajectories, molecular dynamic simulations trajectories. Now, this part we call it the data extracted from known structures, or in this case, even from MD simulations. Then we can do dependently, or to do the principal component analysis, we superimpose the structures, evaluate the covariance matrix, how the individual molecules deviate from the average, and then decompose the covariance into empirical modes. And we can examine the cross correlations, build the covariance matrix. The covariance matrix, you can build it as an n by n matrix based on the fluctuation vector. Or you can think of the components of the fluctuation vectors, each organized in a 3 by 3 matrix. By as you can see here. Now, this type of principal component analysis, now we're going to compare it with A&M predictions. The principal component analysis here applied to HIV transcriptase yielded as the principal component one, the motion shown by those arrows. ProDy will be automatically calculating these things for you, yielding this type of figures or their movies. Now, when we applied to a series of proteins in our original paper in 2009, potential is that the theory and experiments shown by different color arrows agree quite well when we compare the principal components of the ensembles of structures versus the A&M modes predicted based on a single structure in that paper. It's possible to then compare the two. And here is again a compare. You can see the structure here, and the first mode of motion that's predicted by both experiments and simulations. What is interesting to see is that actually the large number of structures determined are nothing else than reconfiguration along this principal mode. So it's as if the structure tends to move along that principal direction at different extents depending on the inhibitor bound, for example. So, in summary, there is a suite of tools that use. There is ProDy, Evol, as I mentioned, for sequence comparisons, DrugWe for drugability simulations, and NMWiz is the visualization software. A newly added module is membrane. Applications in the presence of the membrane, which is also modeled as an elastic network model. You can do mechanical stiffness calculations and compare with AFM data, for example, atomic force microscopy. Or there is this CoMD module that combines elastic network models and MD simulations. Just one example about the membrane before I conclude. You can check that particular module of ProDy, and then you construct in this case elastic network model not only for the lipid membrane protein but also for the lipid bilayer. And then you can see, for example, the motions in the presence and absence of membrane, how membrane remodels the dynamics in a sense, and how the coupling between the membrane and protein keeps the function, the functional motions at least. Here is a very striking example from from a neurotransmitter transporter. This is an aspartate transporter that has been resolved in two different states. An outward-facing state, where it binds the aspartate that is being translocated across membrane to the intracellular region. And inward-facing state is, after binding the substrate, it assumes this inward-facing state to release the substrate to the intracellular region. Now, if you compare the two structures, you can see here that it's a trimeric structure, actually, top view, side view, okay? If you take one subunit and compare what's the structural change between the outward-facing and inward-facing state, here's a movie that has been uploaded by this people. So you can see that actually what is happening is there's a scaffolding region in teal here, and then this transport domain that moves almost vertically. That transport core, let me show you again. The transport core, which is composed of this gray, bluish gray, red hairpin and yellow hairpin, that region just moves, they call it like an elevator-like motion. Once it binds the substrate from the exterior, it moves downwards to release it. So this is a very interesting movement, actually. While the scaffold that is the trimerization domain remains practically unchanged. When we do our calculations, if we do ANM for the protein, for the membrane protein alone, here are the motions that we observe from top, and then from the side when we rotate. This is the most cooperative motion that we observe. Okay. Now, suppose we are now observing the same motions in the presence of the membrane. From it is very similar, although it's a little bit more restricted. But from the side now, you can see this vertical elevator-like motion, which became very pronounced. So this is due to the radial pressure of the membrane, which prevents the opening closing against the membrane, but favors this type of vertical motion. So to conclude, I would like to say that the major advantages of ProDy would be are the ability to visualize very easily the global dynamics, the fact that you can treat very large systems very easily and assess their cooperative behavior. It's very efficient. You get results immediately, and that can help you build new hypotheses. And because we are sampling those large scale movements that are of the order of usually, let's say, milliseconds or slower, these events are very relevant to observables, experimental observables, and you can come up with new testable hypotheses. So I just would like to conclude and by acknowledging contributions that have played a major role in building ProDy resources. And I thank you for your attention.
Great, thank you very much. And if you have questions, anyone with questions, feel free to chat them to me. You can send them in the chat window. The chat window has a caption icon in the bottom, and I can pass those on to Ivet. I had one question, sort of a thought. So you commented earlier that your the principal component analysis revealed sort of conformational substates that correspond well to mutants and other structures, maybe in different space groups and things in the PDB. I was wondering if you had spotted outliers in that regard, things that you might suspect or flag as distorted by, 2-molar ammonium sulfate or bizarre crystal contacts or some sort of other perturbation.
Absolutely. There are always outliers, Jason. And so you don't know why some of, many proteins, when we project them to this principal component space, they form clusters and we can relate them to different functional states. But there are a few outliers always. And we are, so these are case studies, unless there are also some experimental artifacts. I don't know.
Yeah. All right, great. Well, with no questions. Well, with that, thank you again, Ivet, very much. And ProDy is a very cool tool, and it is available. Check out the web-based tools that were also here, and the membrane tools look really great. So, what would you say the sort of focus for future versions of ProDy? Are you headed more toward membrane?
We're going to bigger and bigger systems. Now, what we are doing right now, thank you for the question. That's a very good question. In the PDB, we have large structures, but then there are multiple PDB files that share the same structure. We're stitching them together to obtain even bigger systems, mimicking as much as possible the complexes that are formed in the cellular environment. Because very useful for large systems dynamics, now we have this unique opportunity to stitch together multiple structures and then analyze their collective dynamics. So that is one direction, going to bigger and bigger systems. And the other thing I haven't mentioned is you can use different hierarchical levels of resolution. So we are also exploring the opportunity of, for example, we applied to molecular context, where each protein itself was represented by a single node, and there are more than 400 proteins there. So this is again another direction. Our goal is to move to approach the cellular scale. So uh, that's the future direction.
Wow, great. Well, with that, thank you very much, and I'll follow up with you offline.